I'm just running some buffer through it just so that I have the line all cleared.
Mm-hmm.
So I have, so these are all of the cells, and then these are the single cells that I'm trying to do, and then this is the kind of like the live dead staining channel.
Mm-hmm. And currently it's running the buffer.
Yeah, now just buffer.
Uh-huh.
Alright, so let me just double
For
check one
normalization.
thing. I have live dead CD entry setting equals 780. Let's go. Okay, so now I can run the sample. Microliter at one time. So now I'm gonna do the empty first, like the control.
Perfect.
So the first thing is to also vortex this before we put it on the sorter.
Yeah.
Not sorter, I mean this is flow cytometry.
Yeah.
Okay. Alright. And then click record this. So it will run and record at the same time.
It will run what? Both the antibody and the death.
Yeah. So we have RPE low, the 3.
Is the CD55 measurement?
So this is the CD55 here. So that's our CD55.
Oh, the top one was 0. And the second one is actually the signal for CD55.
This one here is the signal for CD55.
And the left knob was the 0 amount? Or
What?
like the value was 0 of the count, right? Like basically.
The count is
The
number
count is
of
how many cells, and this is
the
the
distribution.
fluorescence, right? So
Yeah.
it was around. This was around 0. Is this 0, right?
0 is here,
Is
all
this?
the way here.
So why was it high over there? Oh, it was like just low fluorescent.
Yeah, it could be that there's not enough fluorescent. Let me just see, put it again as well. So we got this, and then I want to see. Okay, so I want to include this bit here, and then I want so that's RPE, that's the P signal. I want do this.
That
So
is
if I
the
do, so this is live dead.
right.
I'm gonna go to okay, so I don't know why, but there is a low CD55 staining right here, so it could be that we didn't add enough CD55 antibody, but we'll compare with this one now. This is the one with the knockdown.
Yeah. Can I ask about the
Yeah.
plots for a second? So that one you're saying is life and death, and then the x-axis is
So this one,
uh-huh.
the FSC, measure how big the cells are,
I see.
the cell size.
Okay.
And then this is how much it scatters the light. If the cells are dying, they tend to have a lot of vesicles inside the cells, and they tend to scatter the cells more. So anything on this axis tend to be dead cells. But we also have another way to stain the live dead. So this is the live dead here. This is just the cell size. So this is just like measuring how many cells we have.
I see. So the higher it is, the more chance that it's dead.
So these ones are the dead cells,
Got
and
it.
the things that are low over here are the live cells.
Got it. Got it.
Yeah.
Perfect.
And right now I haven't put a gate on the CD55 because I don't know what it looks like with my other sample.
And the other two.
So this one, the BFP, is the vector backbone that contain blue fluorescent protein. Although it doesn't always work very well, but I still wanted to see, do I see any positive staining out of it, and how does it correlate with the CD55 staining?
Okay. Got it. And the top one in the middle. This one.
So this one is to select the cell that are single cells.
I see.
If the cells are double,
Yeah.
the area and the height is not correlating. That means it's not in the diagonal
Right.
axis. So anything that's outside of the diagonal
It's
axis
probably doublets.
are doublets.
Got it. Awesome. Thank you. That's
Okay.
awesome. Great. Thank you.
All right.
Yeah.
Okay. So let me just wash away some of the cells there. So let me see.
You're cleaning it in between the runs.
Where's the buffer? I think maybe this one. So buffer, and also just run that.
Thank you for explaining everything.
Yeah, so you would go back and research
I
it
can do
yourself.
it. Yeah. That would be cool. Okay, so this is the clean buffer, which is why it needs to be empty.
Yeah, so this is just like any buffer that is on there. I just want it to flush
Wash out.
through any of the cells.
What does the number identify? The change in numbers? Is it the amount of cells?
Like the event, meaning like the cells per second is registering, so there's only counting around 200 events, meaning 200 cells or debris in there.
Okay.
So now we can switch to
But we think that it's clean enough.
it. Yeah, like this is just like some of the cells or debris still going through the tubing. And then what is this? Okay. So this is the one edited with CD55. Alright, so then let's go on this one, and then 100 draw, 250 100 draw, 100 to record, record.
Yeah, right. These numbers are much higher. That makes sense. And it also starts with 2 peaks around the same area, though.
Yeah. So I need to see if they come down at all. Okay, so let's then compare. Okay, so then this one. Okay, so you can see there is a diminished
Yeah, the
positive
second
peak,
peaks
right?
are.
So if I put a gate over here, histogram gate, I want oh yeah, so it is this one. So if I want only this one, so only this peak here is positive, right? So if this is the positive,
the one that expressed CD55.
yeah, so let me just customize this. Let me do green. Yeah. So if I only count this peak as a positive, it goes from.
Oh, it aligns them.
So this one is 65%. And this one is 52%.
Okay.
So there are some reduction in the level of staining, although it's not like a massive amount. So one of the things, it could be that we didn't use enough antibody for the cells to stain because we collected quite a lot of events. So let's see, we do this. So anything this way is considered CD55 positive. So if we put the gate on the negative, so this is negative, adding that. So this is only 33% negative. Our other ones is almost half and half. Okay? Yeah. Alright, so then let me just remove the buffer. Okay, so now I can export this.